All right, perhaps we can start. I can see there are already good number of participants. So thank you very much again for joining today's seminar. Can I ask for the next slide, please? So today's seminar is a very interesting one. Perhaps
Sorry,
we,
Sudhir, I must have to share my screen again. At the
all
moment, I
right,
can't
please go
forward.
ahead.
At the
No
moment, I
problem.
can't forward the slides somehow.
No problem, Bob. Take your time.
Yeah. See whether that works better.
All right, please go to the next slide.
There we go.
Here we
Perfect.
go. So sorted out. Thanks, Bob. And as I was saying, that today we have a very special seminar, as you might have seen in the advertisement, that we will listen to Dr. Barbara George-Jaeggli about her life experience and science, talking about crop physiology and linking it with the nature conservation. But before we do that, next slide, please, Bob. As I was saying, it's a great honor to facilitate this webinar. My name is Sudhir Yadav. I'm associate professor with the Center for Crop Science in QAAFI. And before we start our webinar, first thing, as we always do, we would like to acknowledge the country. The University of Queensland acknowledges the traditional owners and their custodianship of the lands on which we meet. We pay our respect to their ancestors and their descendants who continue cultural and spiritual connection to country. We recognize their valuable contribution um, to the Australian and global society. So some house rules, as many of you know that the seminar is scheduled from 12 to 1. And after the presentation, we will hold question and answer sessions. There's no need for you to wait until the end of the presentation. If you have any questions that you want to ask to Dr. Barbara George-Jaeggli, you can type your question under Q&A tab. Please don't use chat function. We are not monitoring that function. So with that, again, it's a great privilege to an honor to introduce Dr. Barbara George-Jaeggli. That perhaps most of us already know Barb, what she like to be called. She work across number of centers and various roles. I met Barb the day I came to UQ for my interview. And during the first interaction itself, I was able to kind of recognize that Bob is a people person, and that's how most of colleagues know Bob. That how well she connect with everybody. Bob has multiple roles. He's with Kofi as well as with Department of Primary Industry. And I think in today's talk, it's a great opportunity to learn from her 30 years of career that how she has blended kind of science and people connections. So with that, Bob, I would like to hand over to you to take us through your journey.
Thank you so much, Sudhir. It's a great honor that you're introducing me because I've really, from the day you turned up for that interview, I enjoyed working with you. And good afternoon to everyone. And thank you to the Coffee Seminar Series to give me the opportunity to present 30 years of, looking back on 30 years of my life and research since I came to Australia, which is in March 1996, I arrived in Australia. So I want to take you through this journey today. I've titled it From Detailed Crop Physiology to Sustainable Agriculture and Nature Conservation, a non-linear career pathway with the big picture in mind. So I've done that. The forward button is always a bit, sorry. I would also like to acknowledge the custodians of the lands on which we meet. I've had the privilege to meet some of the Gidabal people, which is where I sit today at Hermitage Research Station. Yeah, special shout out to them. So I started in agroecology, and basically I've always had an interest in nature conservation, but also sustainable food production, which is why I studied biology, specializing in agroecology at the ETH in Zurich. But when I met Pete, my husband, that brought me to Australia. So the dream of owning our own farm and producing our own food is basically what brought me here 30 years ago. You see me here in front of one of the fields in Europe where some of my work that I did there, bringing back some of the biodiversity into cropping systems, can be seen still now. When I go back, I can see the results of my work. So when I arrived in Queensland, this is the farm we still live on. Of course, that was quite a change. And even so, it's a massive farm in Swiss relations. 60 hectares is not enough to basically live off. So we knew we had to find some off-farm income. And so it happens to be only 30 minutes from where our farm is in Killarney, here at Warwick, is the DPI Hermitage Research Station. And I realized that they do research, and they occasionally have a need for biologists, and basically went there and started as a casual, and then got my first project, looking after a project that was trying to find out what the effects of tree lines on wheat yields in the neighboring paddocks. So that was right up my alley. Pretty much what the sustainability aspects that I'm always been interested in. So I was given a little blue ute. You wouldn't get that today. We hardly have any cars left to travel anywhere, but at this time we had a ute that you would home garage, and I traveled around the Southern Downs to undertake this research. But 18 months later, the project finished, and by that time I was the breadwinner for our family, so I had to somehow look for a permanent job, and there was a job in the barley biotechnology lab going, basically producing double haploids from barley pollen. So while that was not my first choice of topic, again, the big picture was I was the breadwinner, and it was a job in science at all. So it took me a few good years nearly to get isolated microspore culture happening, because that's quite tricky, and we had to set up the whole lab. And as science goes, sometimes the research priorities, as soon as I had it working like a sausage factory and we had barley double haploids coming out of our ears, the government decided we no longer wanted to do this work. So by then it was time for me to revisit my goal to do a PhD, which I had given up in Switzerland to come out to Australia. And David Jordan and Graham Hammer happened to have a PhD in height genes. So they wanted someone to look at the effects of height in sorghum. So David had observed that tall sorghums yield more, and that's not what we normally have come to expect with the introduction of the dwarfing genes in wheat. So Graham and David tasked me with figuring out what the causes of that would be. And a PhD, my first experiments were actually planned for 2002. And as it goes in Australia, that year was a massive drought. So pretty much my field experiments were the only experiment that went ahead at Hermitage. So I'm still thankful to everyone at Hermitage for letting me prioritize this experiment, because in a PhD you only have 3 or 2 years opportunity to do your field work. So I basically used up the last drop of water in that dam for this experiment. So I was introduced to the world of crop physiologists through my work. So learned the tools, so to speak. And crop physiologists, they're pretty reductionist, so a bit contrary to my ecological training. So they look at sectioning everything into what actually yields, what actually leads to biomass. So we've got these elements of water, nutrients, light, and partitioning that people think of. So, and then you break it further down into water is transpiration, transpiration efficiency, and then efficient use of water across the season. Nutrients, about uptake and also using those nutrients efficiently. And similarly with light, you have light use efficiency or radiation use efficiency, and then how much light is intercepted. And you can further break that down into respiration, photosynthesis, and also the distribution of light through the canopy and the leaf area and how long that leaf area is in the field. And then we also think about where does this biomass go? Does it go to the shoot, the roots, or stem, leaves, and ultimately grain, which is the part we're most interested in. So sorry for that, but this is a big part of how I have sort of structured or thought of my research for the following years. So that's why I thought I'd bring that into here. So some instruments, I also wanted to show some instruments that we were using in those days to examine these parts of the crop canopy. And so this here is a neutron probe. Around water, it's neutron probes that you use. So we had a manual weighing system that showed us how heavy pots were to basically work out the water use. So these are our first lysimeter pots. These days we have automated platforms. We had a near-infrared thermometer to measure canopy temperature. And then I also developed this chamber in my PhD, which encapsulated a whole sorghum plant to then work out how much photosynthesis and transpiration is happening. On the side of the nutrients, you have things like a SPAD meter, so you can measure chlorophyll, but you also have wet chemistry, which gets done in a lab. And then for light, we used to use these solarimeters. They're the glass tubes in the paddock to work out how much light gets through right to the bottom of the canopy. And you also dissect your different parts to work out how much biomass is then put on for each light section, basically. And we have a planimeter to measure leaf area. And then photosynthesis and respiration, again, I used my chamber to measure photosynthesis of a whole plant, but also when I covered it up, I could measure respiration. And also we're using a Lycor 6400 to measure gas exchange. And again, a SPAD meter. So then for partitioning, it's a lot of handwork, just cutting plants into different sections and roots sometimes. So then you know where the biomass basically goes to work out your root to shoot ratios, but also harvest index. So what did I find in my PhD using this sectionary sort of approach or reductionary approach? I alluded to earlier that in wheat we had greater yield increases since the dwarfing genes in wheat were introduced. So basically that's what led to the Green Revolution, if anyone here heard of the Green Revolution. And so we got to expect that if you reduce the height of plants, you get more yield. But this seems to be a little bit specific to wheat because the reduction in biomass is pretty much only moderate, but you have a very large increase in harvest index at the same time, which then gives you usually a positive result for grain yield. In sorghum, it seems to be a little bit different. It's a massive reduction in biomass with some of the dwarfing genes, and you haven't got much of an increase in harvest index. So that can be then a negative for grain yield. So that's sort of 3 years of work distilled in one slide, pretty much. So I just got my PhD sort of just before my kids turned up at uni. I thought that was my goal. I didn't want to run into my kids at uni, so I thought I better hurry up and finish my PhD, and it was awarded in 2009. Then these are some of the publications that resulted from my PhD work. But after that, now with these skills as a crop physiologist under my belt, I could apply for a job in Andy Borrell's team. So he's been looking at stay green genes and the mechanism behind the stay green genes in sorghum. And I was successful in getting a job in his team to further look at the mechanisms. So I'm showing here some ways of excluding rain, because of course by then it started raining and when we wanted to do drought research. So these are plastic tunnels that we can grow crops under to exclude rain. And we'll also have these automated roofs at Hermitage that move over the crop once a drop of rain hits the sensor. So that's what we're sort of using to induce artificial droughts, so that we could look at whether these stay green plants, how they did it to stay greener during a dry period, usually at the end of the growing season. So that meant stepping up the number of plant measurements. So from my PhD to this was a step up in how many plants that we've analyzed. Because the other aspect was also we wanted to find the genes behind these traits. So we used pots, but also plants in the ground to take our measurements. And again, in a short one-slide summary, stay green plants, they stay greener because they have fewer tillers and smaller leaves. And if you went back to that physiology diagram, that means they also have less transpiration because they have a smaller canopy. Then that leads to more water left in the soil that they can then use during grain filling. So in the diagram, that looks like this. We've got a little bit of an advantage also with water uptake or transpiration efficiency. And then also nutrients get sucked up at the same time during the grain filling period due to these mechanisms. And you have this change in canopy size. So leaf area and leaf area duration is different in stay green lines versus normal senescent lines. You also have an increased root mass or more partitioning to the roots, and that leads you to larger grains during late season drought. Again, some papers that came out of this work, some of my publications from that, which are still getting cited a lot actually. But then, so that was not too bad finding out the mechanism, but when it comes to identifying the stay green genes, that was a bit more challenging. So what I'm showing here is some of my notes from my Excel spreadsheets. So the green parts show you part of the genome or the chromosomes that have stay green parts in them, whereas the red parts are the senescent parts of the genome. And we're trying to line that up with our measurements in the field. So whether, a plant is actually exhibiting the stay green phenotype in terms of tillering or leaf size or not, and then trying to relate that to the area on the chromosome where we know the genes behind those areas. But as you can imagine, it's quite tricky to always observe the tillering or the leaf size, because you'll have edge effects and environmental effects. So this part was quite tricky. I headed over to Texas A&M twice for a few months to work with our collaborators there, Professor John Mollett and his team. We were growing plants in their glasshouses and doing similar measurements, but also included gene expression studies in those experiments. So they were experts in that aspect. That was my opportunity to go to where, Norman Borlaug, so the famous Green Revolution researcher. So that was a really memorable time over there. So in the end, it was more circumstantial evidence to say, rather than prove which genes that were driving the stay green phenotype, because we didn't actually have any genes that polymorphism in the genes. So, but we knew that they were close to genes that are affecting canopy. So these auxin efflux carrier were our prime candidates. And also Emma in her work found that indeed these auxin efflux carriers or PIN genes were co-locating with the areas in the chromosomes in the genome that were related to stay green. So I call that circumstantial evidence that the stay green genes have something to do with these PIN genes. And we also later, Andy and other people expressed these PIN genes or induced them, and they indeed got the same sort of phenotype, being smaller leaves and smaller, lower tiller number. So now again, with further skills under my belt, I was eyeing off a project in QAAFI that was going off in QAAFI, because this previous work was all done under DPI. But QAAFI was, some people entered this Translational Photosynthesis Center of Excellence, and I was always interested in photosynthesis, and also had met Suzanne van Kamera, people who work in photosynthesis earlier, and thought, wouldn't it be great to work with people like that? And here was an opportunity. So I applied to do a part-time postdoc in this project. And again, looking at what we're actually trying to do. So they were saying that most of the breeding efforts have gone into increasing grain yield. So the harvest index has been driven up. Again, that's very true for wheat, probably less so for sorghum. But if we wanted to have another green revolution, the green revolution, was actually driving this increase in harvest index. But if we wanted to have another yield revolution, we needed to look at biomass. And this is where I was very familiar with all these parts of what leads to biomass. So I could again contribute by taking measurements to find these mechanisms. But we needed to step up the number of measurements again. So from hundreds of lines I was looking at in stay green experiments, we now wanted to go to thousands of lines. So we needed a revolution to make that happen in phenotyping. Swiss people are good at revolutions, so I thought I'd be perfectly fitted to lead that work. I'm showing here some of our sorghum trials, but also some wheat high throughput phenotyping trials in Yanko that people were working on through the Center for Excellence in Translational Photosynthesis. So for me, it meant first to take thousands of single plant measurements by hand in the field, me and my team. But then we later got some help from this gecko. So we had this old tractor, a spray cube, that we loaded with different types of sensors. So from infrared to LiDAR to hyperspectral sensors, we put everything on that we could. Most importantly, GPS, so that we knew where in the paddock we were. And of course, to make sense of any of that, we also had to accompany that by over 4,000 hand harvested biomass samples, so that we could then develop algorithms. So it's a big team of people involved in all of this work. We're also using drones to take measurements. And so we thought, we had solved our problem. We can now phenotype thousands of lines in the paddock. But of course, the next challenge was, what are we going to do with this big data? Because only one of the instruments, the nano head wall, actually collects over 10 million values per square meter. And most of our trials were around 2 hectares. So you can imagine the data that comes off these missions when we collect data with drones and the tractor, the gecko. Each of the drone flights actually in itself gave us 32 gigabytes of data, which I calculated as equivalent to the information in 38,000 copies of Harry Potter, The Deathly Hallows. So you can imagine how much data comes off these missions. So by now, the sorghum team tells me they actually have collected 62 terabytes of data, which is stored on our servers. what we also had to develop was a pipeline to go from raw pixel values to then something that breeders can use or sorghum physiologists can use. And this pipeline work has been published, and it's also been used in other crops now and has led to further investment such as hay gain, which is a project you can hear from in next week's seminar, I believe, from Anna Coltenow. So traits we have developed algorithms for in this work, and we've got some static ones on the left-hand side here. So the plant number, head number, leaf area or cover, biomass, yield, plant height, light interception and light distribution, radiation use efficiency, canopy temperature, chlorophyll content, and photosynthetic efficiency. But because we're taking measurements weekly now, or even more frequently, you can also look at dynamic traits. So your phenology, your development of the canopy, your growth rates for biomass, for example. You can look at things like leaf area duration and get back to stay green, looking at stay green, or also lodging. So it really opens up a lot more than just looking at yield or some quality traits that maybe used to be looked at in the past. So the way we envisaged to use all these new technologies and resources, we're going to use crop models to have prior knowledge on which traits are actually affecting yield at the canopy level. Then using these phenotyping resources that we've built to screen the trait in diverse mapping populations, and then bringing that together. So integrating the phenotypic information with genotypic information to identify the genetic loci or the areas on the chromosome where those traits are located. And so that we can then select for in breeding populations and translate that to yield improvement. And ultimately, this knowledge can also be transferred from sorghum to other crops. So this working in the Centre for Excellence for Translational Photosynthesis was actually a great opportunity for me, also for my personal growth. It opened up opportunities also to meet giants in photosynthesis. So on the left here you see Hatch and Slack. So they're two researchers that used to actually work in Brisbane in sugarcane, and they discovered the C4 photosynthetic pathway. So sorghum uses the C4 pathway. So they're certainly legends in that type of research. And I also met Tony Fisher, people like that, and had the opportunity to go to an ecophysiology workshop where we deliver training with Suzanne van Camera and Graham Farquhar. So again, they're legends in photosynthesis research. So I actually got to travel to China multiple times through these projects, which was quite eye-opening. So the second time was the second Asia Pacific Plant Phenotyping Conference in Nanjing. And I also visited Anhui Agricultural University in 2018. And then an international phenotyping symposium in Nanjing, and I visited the Cotton Institute of the Chinese Academy of Agricultural Sciences in Anyang. So they're very memorable trips. Resulting publications again from this work. We've had a few papers that come out. One of them is a Tansley review, so that got quite excited quite a few times. So great opportunity to be working with people like that and be able to publish this work with them. It also gave me opportunities to supervise students because we've had a 5-year project, so you can actually see a PhD or master's through. So one of my first students was Gedika, and then Xiaoyu. So you can see Xiaoyu here taking hundreds of photosynthesis measurements and other students that I had. So that was, I enjoyed that part. And again, some resulting publications from the students' work. So just acknowledging these phenotyping resources, we could only basically develop them and buy these expensive pieces of equipment because we had funding from the Australian Research Council through the ARC Centre of Excellence. Also, the University of Queensland gave some money, Bill and Melinda Gates Foundation, and the Queensland government. Without that sort of financial input, you could not accumulate these types of resources that can then be used in further research. So it's been a great project in that regard as well. And obviously it took a lot of people to work on this. So it was a great team of people. These are just the people from the sorghum part of things. There were also people working in wheat that I haven't mentioned here. So I have some other roles, as Sudhir also alluded to, and have taken opportunity to make agriculture more sustainable through some of those roles. So one of them I've been very passionate about is raising the next generation of scientists. So for more than 30 years now, we have been running a plant science competition out of Hermitage, and we've reached hundreds of thousands of school children and get them to come to Hermitage and learn about the work of a researcher or anyone else working in our teams, so they can envisage a career in agriculture, because without that, we're not going to be sustainable. I've also been always very passionate about equity and diversity, so have worked on different committees and working groups to enhance the opportunities for women, but diversity in general. Have been on different forums as well. And also passionate about mentoring early career researchers. So I've actually led the first mentoring program we had in QAAFI, and then also through my theme leader role, had further involvement with EMCRs, or early career researchers. So the QAAFI theme leader is also a role that I really enjoyed. So when Matthew Morrell introduced themes in QAAFI that are cut across the different centres, I was very keen to put my hand up to lead the sustainable agriculture theme, and Sudhir later joined me in that theme as well. And that's been really also a great opportunity to connect with people across UQ and other universities. One of the more major successes was the funding for a cooperative research centre for SEER net emissions from Australian agriculture that got together. So that's certainly something I really enjoyed being part of. But we're also part of a climate change network, research network across UQ that puts on different events, bringing researchers from different disciplines and areas together, working on climate change. Again, something I'm really enjoying. And then there's also a lot of tries that might not necessarily lead to funding, which that's how it goes in research. So I'm just showing my folder structure here. That's all folders associated with projects that we've been involved with and talking to people and trying to get funding for, but not necessarily successful. So that's part of challenges in research. I'm also taking sustainability into my Hermitage Centre leader role. So when we turned 125 years, we took the opportunity not just to celebrate 125 years of research at Hermitage, but also acknowledge that there's been 60,000 years of sustainable food production in this area going on long before the white settlers arrived. So that was a very successful celebration where we came together with the community to create some artwork that's now installed in front of the Hermitage. We also took part in a DAF Carbon Challenge in 2020. So we were campaigning for some solar panels at Hermitage. Unfortunately, COVID, it stopped many things, gave that a bit of a break. But this year we actually just got our solar panels installed at Hermitage. So a fair bit of our power can now be delivered through solar panels, which is a great outcome. So looking back, things that have changed in 30 years since I started at Hermitage. We have email now. So when I first started, believe it or not, we didn't have any email. I don't know what we did all day, but we certainly didn't read any emails, but they got introduced not long after I started. Also, diversity is something that has changed a lot. So I was one of the only technical women. There was only one more lady who was working in a technical role. Other than that, they were all basically men working at Hermitage. And that has certainly changed since then. We have a lot of women and they come from all over the world. And we also have more diversity across people. The male colleagues, they also come from all over the world. So that is something that really has noticeably changed to the better. It's been really great to work with these great people from all over the world. Then funding, as I already alluded to, is harder to get these days. And more research is being done via PhDs rather than long-term teams. Especially in DAF, we used to have long-term teams of researchers and technicians that became quite skilled. But unfortunately, these days, research seems to be a lot shorter and people only get fixed-term contracts, which makes it really tricky to develop an expertise in something. And certainly the number of breeding lines that we can now evaluate has increased massively thanks to enabling technologies. And I would like to point out two of those fundamental technologies. One is the 454, also called as next-gen sequencing. So it's a way of analyzing the genome of our lines. So we're now able to have genome information for hundreds of lines at quite a fast pace. So that's certainly something that enabled a lot more faster breeding and greater populations. But then on the phenotyping side, it's basically the GPS that's been the technology driving it, because you can imagine if you're trying to run your tractor through the field, you need to know, or even your UAV, your drone, you need to know where you are, which plot you're looking at. So to me, that seems to be the most important enabling technology in stepping up and scaling up our numbers of lines that we can screen in our field experiments. Other things that have changed in 30 years, unfortunately, carbon dioxide in the atmosphere has risen from about 360 parts per million when I started at Hermitage to 429 in 2026. So I actually noticed that because I used to hear about climate change at uni, of course, and also we learned that the atmosphere contained 330 parts per million of carbon dioxide. And when I started to do my photosynthesis measurements at Hermitage, I thought my instruments were broken because they were closer to 380 when I first started. But I soon learned, yeah, this is really driving home what's been happening with carbon dioxide. And ever since, they've gone up by 2 or 3 parts per million per year. So to lead us at 429, which is one of the reasons we believe in planting trees on our farm. And the trees, the ones we planted 30 years ago, they've also grown considerably in the last 30 years. So that's one thing that's changed to the better, I feel. And I'm also now a grandmother, mother of 3 lovely girls. So big change in my life as well. Our farm is bearing fruit. So all these fruit trees, nut trees, things we've invested in and put in are now bearing fruit. And quite often we look at our meal and about 90% of the produce comes from our farm, which is a really great feeling. Then there's also some things that have not changed in 30 years. And I actually would like to have some more time to, because my interest in platypus research that I've had ever since I first saw a platypus when I came to our farm has not gone away. So I would have loved to study platypus, but I had to be realistic and see the bigger picture that a job in agricultural science is better than no job. And at that time, there was not really any funding that I felt for this type of research. So I've been able to have my career, but study platypus as a hobby on the sideline. So every 6 months, we organize some surveys with the community, together with Wildlife Queensland and Kalani Bush Care, to keep an eye on the numbers of platypus, and because Queensland is lacking in a lot of conservation data, and without that data, we can't protect our natural resources. So this is my contribution to help with some of that data. I'm also volunteering for Landcare and undertake projects in sustainable agriculture through the local Landcare that we have in Warwick. So going forward, I'm hoping to work part-time to have some more time for these interests and get back to my initial interest of conservation and agroecology. I'd like to acknowledge so many people and funding bodies and my family. As Sudhir alluded to, I really enjoyed working with people and have met so many across my different roles and in our teams. And it's been a real pleasure to get to know everyone and working towards a common goal. I've even had the privilege of working with my own husband. He was a technician in the sorghum team, so at times our work coincided, and we're up in air collecting seed for the sorghum team. Yeah, so that would be all I had to say, Sudhir.
Thank you very much, Bob. This was excellent and beautiful, and thank you very much for sharing kind of glimpse of your 30 years of journey. Yeah, it was fantastic. I had so many questions in my head, but perhaps we can start with the questions that already started to pop in Q&A. So we already have a question there. It's the question is that thanks for the overview of your fascinating research. You mentioned about your visit to Texas A&M. So did you have contact with the perennial sorghum research team there? And the second part is, might that be relevant to Queensland or elsewhere in Asia or Africa?
Okay. No, I'm not sure that I had contact unless people were working on perennial sorghum then that I didn't realize. But no, but I do think perennial sorghum has got some opportunities, especially with regards to sustainability. Obviously, you don't cut the crop down every year, so you can keep that root mass in the soil, which is something that's very beneficial. So yeah, I do think, and I think people are looking into that as well, and so has the sorghum team at times as well.
Good, thanks. I just want to remind audience that you can put your any questions or comment in Q&A tab, and we will take as many as we can. While we are all right, so I think we already got. I'm not getting time to ask my question, which I was also very much willing, but I think this is very interesting. That has your attitude to snakes changed over the years?
Oh, we're just talking about snakes at the Monday morning meeting this morning. Yes, I think it has. So if you remember those solarimeters, those glass tubes that I showed you that we were using to measure light interception through the canopy, they had to be cleaned every week so that obviously they collect the right data. And one time when I was doing this job, I put my hand down to clean this solarimeter on the ground, and next to it was this brown snake that actually had a go at my hand and just stopped about 10 centimeters in front of my hand. And we both sort of retreated and looked at each other. And then I thought I better finish what I was doing, take some more measurements while I know where the snake was, before I run out of the paddock and then have to come back later. So I think, and in that time was probably about 15 minutes that the snake and I spent together in the paddock. I think I learned that they are very forgetful. So if you don't move, they do not notice you there at all. But if you move, including stomping the ground, at least the brown snake came forward and wanted to know what I was. So main lesson is stop still. And also, first aid, there has not been a fatality in with snake bites in Australia for a long time because our first aid is so efficient at if you don't move after you've been bitten and you apply pressure bandages, you'll be fine.
Good. So the congratulations message are flowing in for your fantastic work over the years, Bob. And we have more questions that again people are appreciating your presentation and reflection of your experience. There's a question that despite the project being defunded, have you heard of any work continuing regenerating barley plants from the pollen?
Oh, yes, it was not funded by the government, but there are other labs because we were outsourcing it to other labs. So, yeah, that work is still going on in other places, just not with the Queensland government.
And again, that's sort of reflecting back in your presentation that looking always towards the next stage of taking that work. I think there's a question about your how do you see ag research? You talk about kind of last 30 years, and how do you see for the next 30 years that having no email to so many emails, what else you see changes in next 30 years?
Oh, that's a hard one. I mean, obviously, everyone talks about AI and that we will be all out of a job. But I do think that there is still some physical things that we can't change. You know, the world still has got physical limitations, which will mean there'll be always a job in growing food that can't be done totally by AI. And obviously also the resource constraints that are coming our way will mean that some of the research will be looking at how we can make plants more efficient. And I think Australia has a massive benefit here because we have been able to learn so much about growing plants with little water, so that the rest of the world can probably learn from us in terms of dryland agriculture and how you really optimize that system. We have a lot of experience in climate forecasting, but also then using every drop in the paddock as efficiently in your cropping system as you can. So I see a role for Australia in future to the rest of the world as they're catching up with drier times.
Yep. And that's the prediction for this year that it will be quite dry. So yeah, we do need to do some catch up. Just slipping in one of my thought, Bob, that you have been known as a strong advocate and supporter of early and mid-career researchers, and you have seen a lot in last 30 years, sort of lot of uncertainty and changing expectations. And it was, if it was not uncertain before, it was a lot now, and it will be more uncertain in future. So, what, from your experience, what advice would you give to early career researcher about staying purposeful while still being open to that those unexpected opportunities? I think in your career you have seen a lot.
Mm. Yeah, so for me, it's basically around not being too rigid, but still have your values. So have your core values that you want to have, impact on sustainability, for example, but you can apply that with different roles. As you've seen, I've started with ecology, conservation, but couldn't really find a job in that area. But every role I've taken on, I could sort of see the bigger picture and go, well, that actually ultimately leads also to a more sustainable world. And taking pressure off nature if we have more yield in our crops, for example. So staying true to those values would be one way of being a bit more relaxed, exactly what you're doing. So if you're not really so focused, I just want to do this one thing, that might not work out. But if you just concentrate on the values behind it, then you might be able to bend your career path. And the other thing, which is probably not what I have done well, but if you can develop some expertise or unique skill that people are looking for, obviously. So it needs to be generic enough, so it can't be totally in a niche area that people are not going to fund anymore, that sort of thing. It needs to be generic enough so you can apply it across a few research projects, but it needs to be something that people come looking for you to collaborate with, I think.
Yeah, no, very good advice. Thanks, Bob. We will keep blending life experience and science. So there's a question about what is the relationship between the stay green trait and late maturity in sorghum. That's the first part, the relationship between stay green and late maturity. And the second part is, and from an evolutionary perspective, is stay green a common ancestral phenotype in sorghum landrace?
Yeah, so if I start with the second one, well, I'm not the breeder, but that's pretty much most of the sorghum is nearly perennial. So it seems to be it's not like wheat or maize that dies after, in the grain field period. It really has a bit of a perennial characteristic left. But in terms of the maturity and stay green, they don't have to be go together. They're not intrinsically linked. So, for example, in our experiments, we had lines that had exactly the same maturity, but one was stay green and the other one was senescent. So yeah, that's not actually linked. But of course, the leaf area. So if you're looking at the leaf area after flowering, that is actually there longer, so you have more green leaves for longer during the grain field period, which then lets the plant photosynthesize longer and puts that into the grain. But the overall maturity is not necessarily different, if that makes sense.
Good. So we will perhaps now take a life experience question. So you talk about sort of next 30 years, what could evolve. There is a question about that if you look back at your 30 years of career and had a chance to remake a decision, what would be that?
Probably what I said before, get myself a unique skill. I'm a bit of a generalist, and my skill is more, having an interest in the big picture sort of thing. So I probably failed to develop some really expertise in one technique that people come looking for to me. So maybe that's what I would change. Try to find something that I can then offer to collaboration teams.
Good. So yeah, there is a very good blending of questions, science, life experience, and now to community service. So you talk about platypus monitoring. Did you see any change in the number over the years?
At the moment, we've had some good years. So since we've been doing it, so we've been doing it for 5 or 6 years now, and we've been lucky enough that that was sort of post a big drought, and the numbers have been increasing, and they've been stable in the last few years. But I'm dreading the next drought. So obviously, once we get the next drought, populations will crash. And the problem is in some river systems, they don't come back. Once the water comes back, they not necessarily go back in there. So that's a real threat for platypus. Drought is something they can't really deal with because they need water to find their food. Yeah, so the last few years we've been lucky, good numbers or stable numbers, but we'll see what happens when we go through another drought.
Yeah, and as you said, that I think the most important thing is that making this kind of a continuous monitoring exercise rather than one time off, so we can
Yeah.
understand what is happening and take actions. More messages are flowing to congratulate you, Bob, for not only for excellent presentation today, but also again 30 years of service. I don't see any particular. Here we go. Perhaps we can take it as a last sort of again appreciating your presentation and service, Bob. And the question is that what unique skills specifically you would now recommend for early career professional wanting to enter the ag industry?
Well, maybe 2 years ago I would have said, coding, but that's already been taken out by AI. So it wouldn't be that. I think get back to the bigger picture, maybe we need more people who can really link things, like the supply chain. I mean, it shows us how vulnerable we are. So specialists in really seeing how things are connected. People like yourself, you look at LCAs and that sort of thing. Yeah, I think that would be a skill because everything obviously has a cost and a benefit. So we need to be really aware of our choices of those costs and benefits. And I don't think we have been in the past in terms of where we, put our money in agriculture or what we produce. So maybe that would be a good skill for someone.
And now again, good suggestion. I think that's perhaps I can see the last one. But Bob, again, thank you very much for this excellent talk and sharing a lot of learning and how your career has evolved. I like now and then when you were saying that big picture landscape also change, and so many times you prioritize work while at other time you prioritize family, which is sort of again tell a lot about how one need to be kind of remain aware about that what are the priorities. It's not only about one skill or so looking at that bigger picture is very important. So thank you very much for sharing your experience, and all the best for next stage of your life and career. So perhaps we can wrap here, give everybody, it's already dot one. So if we go to the next slide, please. So I think that's sort of end of this seminar. And if you haven't signed up for coffee seminar, you can scan this QR code and sign up and get familiar about the upcoming seminar. So with that, thank you very much for everybody for joining today.
Thanks everyone for the
Bye for
excellent
now.
questions, and thanks Sudhir for chairing.